this thing on? Because it's getting ready to be on.
Hello everybody, and welcome to Bell Ringer. My name is Greg. Your guest name today is Sarah Bowman from the Hauptman-Woodward Institute in the heart of the Buffalo Niagara Medical Campus. Sarah is a scientist running the crystallography lab and structural biology. We have a great and important conversation about the work they're doing on the COVID-19 pandemic and the process through which will lead to an eventual vaccine, how that path goes through Buffalo. Thank you so much to Sarah for her time and her work, and each of you for listening. Thanks, guys. So for those that don't know, tell people a bit more about Hauptman-Woodward Institute and some of the history behind it.
So Hauptman-Woodward has actually been around for just over 60 years. It's really a thriving structural biology hub. And structural biology is really the study of how things look at a very small, detailed level. It was founded with the financial support of Helen Woodward Reavis, and has, really been doing a lot of fantastic work for the past 60 plus years in predominantly structural biology. We, of course, had one of our directors before I actually started, Dr. Herbert Hauptman, who's a Nobel Prize winner from right here in Buffalo, New York. And I guess that's really representative of the type of the level of scientific kind of effort and work that happens at HWI.
Yeah, I actually was in the building for the first time a couple of weeks ago. I interviewed somebody in the lobby and I saw the Nobel Prize right there.
Yeah, it's pretty awesome, isn't it?
Yeah, amazing. And
right here, the etching along the glass there is all equations from Herb Hauptman.
Really?
The equation, yeah. And so in the very front of the building, there's all these really beautiful etching of diffraction patterns and the equations that he actually got Nobel Prize for. And so it's an amazing thing. If you're a kind of a geek like me, that's really awesome. So
that's amazing. Yeah, for those that are listening, not from Buffalo, right, kind of in the heart of the Buffalo Niagara Medical Campus, surrounded by a ton of relatively new and beautiful buildings. So there's a ton of innovation going on kind of in that research park.
Yeah, it's great. Actually, the HWI building, which is so unique and beautiful, right? Was actually the first building that was part of the first new building that was part of the new Buffalo Niagara Medical Campus. And HWI is one of the founding members of the BNMC. And so, and it's really a vibrant area with the medical school right there. We're directly connected to UB with the computational resources there. And then Roswell Park is right across the street. And then there's all the startup things that are happening everywhere there as well. So it's great.
Yeah. So within Hauptman-Woodward, you're charged with running or directing the Crystallography Center. Is that correct?
Mm-hmm. Yep.
So tell people what that is a bit more specifically.
Okay. So the Crystallization Center that I direct is a high throughput crystallization screening center. And so the first question is, what is crystallography and what is crystallization and why do you need it?
Read my mind. That is the first question.
So, sorry. So the answer to that is when we are trying to understand what different molecules are within human body and within viruses and within bacteria that, for instance, cause disease or mutations or variations in proteins, which are kind of the molecular building blocks of cells, we need to try to understand what they look like. And so one of the ways that we do that is actually using a technique called X-ray crystallography. And in that technique, what we do is we shoot X-rays at our crystal sample. And so the first thing we actually have to do is make the sample that we have actually form into a crystal. We have to coax that to happen somehow. And it turns out that this is a huge bottleneck. So there's a worldwide database that is called the Protein Data Bank, and it has the over 160,000 different structures that have been solved. Most of those structures have been solved with X-ray crystallography. And so most of them have gotten to that crystal stage. But because it's a huge bottleneck, and it's one that typically takes a lot of trial and error and experimenting and trying to just figure out how to take this pretty precious sample that you have and actually make it go into a form that will then that we can take the experimental data on to actually be able to tell what it looks like. And so about 20 years ago, February 2000, they started this high throughput crystallization center, and it's really been the center has been at the forefront of doing a lot of the research into how these methods actually work and how to get macromolecules to crystallize in that entire 20 years. And so what happens is that people from all around the world actually send their samples to us, and we have a lot of different robotics and imaging instruments and specialized equipment, and we've been running for 20 years, and so we have a lot of expertise. And we set people's samples up for them, and then we have a very specific kind of experimental technique that we use to figure out what conditions will crystallize, in what conditions the samples will crystallize. And so we try to kind of remove that barrier for people who are trying to understand the particular samples that they're working with and the proteins that they're working with, which are really important for kind of understanding disease and just figuring out what things look like at all. So that's what we do. And then we send them, we have this experiment that actually sets things up for about 6 weeks. And then we monitor the conditions for 6 weeks to watch when those crystals grow.
Right. And then, obviously top of everyone's mind is the COVID pandemic, and, to be able to put that process into practice, for something that's so relevant to everybody. How has the center played a role in that, and some of this science or research with the coronavirus disease?
Well, so that's a great question. So I think that one of the really interesting things is that the scientific community as a whole has really dropped everything and just been really focused on trying to understand the virus and the different pieces and parts of the virus in terms of the, kind of first line responders being able to figure out how to even treat people who get this disease, the epidemiologists who are trying to understand how it's spread. And then what we do is structural biology. So what structural biology gives you, like I said, is a sense of what things look like at a very small level. So if we think about like the overall size of the SARS-CoV-2 virus, so that variant is around 100 nanometers. So that's extremely tiny, and that contains everything that's in that virus, right? And so if we want to try to understand, for instance, how to develop a vaccine to stop the virus from entering the human host and the human lungs and all of that, or if we want to try to understand how to stop some of the proteins that are responsible for the kind of infectivity and the fact that the virus is able to replicate within the human cells, then we need to be able to see what they look like, these things, right? And so the virus itself is really small. The things that actually make up the virus are therefore even smaller. So to be able to look at these things, it's almost like looking at things that are virtually invisible. So if we were trying to see these things with just the naked eye, that would never be possible. We see these big pictures of the variant where there's like the big sphere and it's got like the red spikes coming out of it and the other kind of colored things. Well, so that red spike is a protein, and so to kind of really get a good sense of what that protein looks like, to be able to design a vaccine that will help to stop it from being able to bind to the human cells and actually then release the variant inside the cells, means doing structural biology. Okay, so that's kind of our basic our basic thing there. So what we do, what we've been doing in the Crystallization Center. So obviously in March we shut down for normal samples. So we typically receive samples from all over the world, like I was saying. And what happened was around the same time that New York was shutting down initially, we were getting contacted by researchers who were already working very quickly on SARS-CoV-2 proteins. So these are pieces of the virus that are important for how it works. And saying, can you please screen our protein samples for crystallography to try to figure out what things will crystallize. And so we were able to kind of put practices into place to make that happen and safe for kind of our group to be able to be in the building. Everybody is staggered and really only working kind of one person at a time. And we're still trying to do that, even though we're, kind of ramping slowly back up, obviously. But we've been very successful in that. We were able to get National Science Foundation funding for rapid response to be able to fund these experiments for people who are sending things to us. I can tell you that, so we work with a lot of confidentiality until the results are actually out. So I can tell you we're working with samples from many different states from all across the US, and we've had inquiries from international labs as well. And that we so far have already participated by being this kind of finding the initial conditions for a particular protein in the SARS-CoV-2 that is called the main protease. And so this is a really critical drug target. And so our users are from Oak Ridge National Laboratory, and they have been able to solve this structure very quickly. And in part, it's because they were able to find the crystallization conditions very quickly by using the high throughput crystallization screening center. So that's kind of part of what we're doing. We're one of 3 groups at HWI who actually got NSF rapid funding for SARS-CoV-2 projects, and so we're very active actually at HWI in 3 different areas, working on trying to kind of confront this. So
yeah, and when you talk about the ability to speed up the process through this crystallization, I guess as an example, like, what kind of time frame difference does that create for people, or, for your end user, for the research?
It's pretty substantial. And I will say that the other thing is that the structural biology community has also really come together and been working very hard together to release their data and release information very quickly. So, it's funny because I was actually at a conference last week virtually, and we were talking, it was a structural biology conference, and we were talking about the just amazing response time that has occurred. So I was telling you about the protein data bank, which kind of holds all of these structures. So normally, once something new is discovered and you're trying to get a structure, it could take years to get. And we had the first structures of some of the proteins within months. And at this point, this is, of course, COVID was really identified in December of 2019. Here we are in August 2020. And, I actually just looked the other day, and we have just over 300 structures of some of these proteins. Now, there's only, 28 proteins in this variant. And so there's structures with lots of different drugs bound, or potential inhibitors, or things that might help us to design better drugs to actually stop various proteins from functioning. And so it's actually been a really amazing time to kind of be a scientist and to be in structural biology, to be working with this stuff.
And when people talk about a path to an eventual vaccine, your work at HWI, and I guess structural biology, someone has it in the name, structure is one of the first steps on the path to getting there, right?
It typically is, yeah. So so in terms of trying to figure out how to design a vaccine that's actually going to be effective, there's of course always there's all manner of kind of clinical trials and things that we hear a lot about. And before we kind of get to that is typically information about what the thing is that we're trying to vaccinate against. And so in this case, the spike protein that we were talking about earlier is a key kind of vaccination target. And so having a sense of what it looks like, and what all the different pieces and parts are, what it looks like when it's closed up and on the virus surface, what it looks like when it's open and actually coordinating to a human protein. These are all things that are really important for us to be able to kind of say, okay, well, what would be a potential thing we could use to vaccinate against? And then the other thing is the treatment, which is, so there's vaccines, which are kind of preventative, and then there's treatment methods, which are more treatment. You've been infected with COVID-19, and what's going to be a good way to actually stop that infection before it kind of proceeds. And those are also really impacted by structural biology. So there's an entire field called structure-based drug design, where having the structure allows us to kind of design drugs to go after just that structure, but not structures of, say, your human proteins, because obviously we don't, it's always a delicate balance to make sure that what we're putting in to kill the virus is not going to harm the human. So
yeah, and you have all of this obviously very intensive work going on. Simultaneously, you're under a bit of a construction project with the Cryo-EM Center. So
right.
Tell everybody a bit about that and how that's gone, balancing the two with probably, like you said, shutting down a bit in March, coming back, working safely, but also constructing this new project. And I think you guys are maybe in the process of wrapping up.
Yeah, the construction is going really well. So as I mentioned, X-ray crystallography is a major technique in structural biology, and I would say that the other really amazing technique that's getting a lot of that's kind of the cutting edge right now is cryo-EM. So that's cryo-electron microscopy, and what that requires is essentially having a really big microscope to be able to, again, look at these really small molecules. And so it's a very similar kind of thing. We have to take our very small molecules and look at them with something. And so we are in the process of finishing construction for the Cryo-EM Center at HWI. And that's super exciting for us as structural biologists because, especially because, for me, it might be a little selfish, but for me, it's really exciting because the Crystallization Center is on the second floor, the Cryo-EM Center is on the first floor, and we will have a lot of kind of back and forth talk because you never know what the best technique is going to be to actually solve the structures of your particular kind of molecule. And so it's really great to be able to kind of have both things. So the construction has been proceeding during the shutdown. And in fact, it's been really interesting because when you only go in every few days, things proceed very quickly. And I believe we should be getting our Cryo-EM installed, our first Cryo-EM microscope installed sometime quite soon. We've hired a new Cryo-EM director, Dr. Shuxing Zhang, to kind of head the center. And so it's really great. And the other thing is that we actually HWI runs a particular synchrotron beam line actually out in Chicago at the Advanced Photon Source, which is called IMCA. And so between the Cryo-EM Center, the IMCA beam line, which is really specific for a lot of pharmaceutical companies to be able to use, and the Crystallization Center, we actually have all of I would say, many of the tools that we really need to actually be kind of the hub for structural biology in in the U.S. So.
Right. And that actually transitions nicely into what I wanted to talk about next. And we somewhat got into it at the beginning, HWI's place on the Buffalo Niagara Medical Campus. But, more macroscopically, I don't know that many people, when they hear Buffalo, think about a lot of the work that you just talked about, being located here. But there really is, we really are, or at least have the potential to be a hub for, life and health sciences.
Mm-hmm.
Talk a little bit about your experience, not just at HWI, but, within this community, both, within close proximity around you. You mentioned Roswell, but, kind of Western New York more widely.
Well, so I actually did my graduate work at University of Rochester. So the first time I came to Buffalo was actually for a conference that was held at the HWI building, which now I was actually doing a different experimental technique at that time, but it's a really beautiful building, and it's a really wonderful place. You walk in and you really enjoy being there, and you want to kind of go to work every day. Within, I think that in some ways you could think of HWI as being a little jewel in Buffalo that a lot of people in Buffalo don't necessarily know a lot about, but that within kind of the wider scientific world, it's pretty well known in terms of structural biology and the work we've done. And in part that's because the Crystallization Center has been kind of up and running for 20 years and is kind of known as a very innovative and resource for people to be able to use. And in part, it's just because there's a lot of really good science that's happening at HWI. And so, I was at this meeting last week in structural biology, and everybody there knows HWI. And, I did my postdoc work at MIT, and people there know HWI. And it's very strange because sometimes you're in Buffalo and talking to people who are not scientists, and they say, "Oh, I don't know what HWI is." And so I think it's really great to kind of be part now of the BNMC, which I think is expanding in really fantastic ways. So I've been at HWI for just 3 years now. So I watched the medical school being built. Essentially, it was kind of a shell when I arrived in Buffalo, and I've also watched the kind of community in Buffalo, the scientific community, just become, I think, more engaged with one another. So I have collaborators and coworkers who are at Roswell. I have collaborators and coworkers who are at University of Buffalo at the Medical Center. I have an appointment in the Department of Biochemistry at the med school as well. And I think that also translates to having a lot of interactions also with the other kind of schools in the area. So one of the great things about the Cryo-EM Center is we will have the only Cryo-EM Center in Western New York, upstate New York, so it's great because we get to interact with the scientists who are here in the Buffalo area, but also at University of Rochester, RIT, kind of down into the southern tier, and just all over the place. And so, and I think again that because HWI is such a kind of a important contributor in structural biology, and that people really know a lot of the scientists who are at HWI kind of in the larger structural biology community. I mean, we end up working and interacting and collaborating with people in California and in Chicago and in, all over the place. So
we're going to we're going to help spread the word through this podcast here in Buffalo.
That's great.
Well, thank you so much for your time today and all of your incredible work, always, but especially over the last few months during this pandemic. Obviously, everybody is, so grateful, and Buffalo is certainly lucky to have you and everyone at HWI. Before I let you go, we have a couple hard-hitting blizzard round questions. I'll toss a few at you, and then I'll let you get back to it. If you were a flavor of ice cream, what would you be?
Coffee.
Book or TV show that you'd recommend?
I don't really watch TV, so I can't recommend any TV shows at all. However, let's see. Especially right now because of the pandemic, I've been reading Spillover, which is a nonfiction book about essentially the prevalence of various viruses in the world and their spread into human populations. So, as a kind of what's happening right now and getting a bigger sense of it, I think I would recommend that. Great.
Text or phone call?
Text.
Bills or Sabres?
Bills.
I know you're newer to Buffalo, so that was a tougher question. Hiking or skiing?
Neither.
Okay. And then final question, most important one, chicken wings, drumstick or flat?
I'm partial to kind of vegan treatments of kind of chicken-like things. So yeah, I'm not sure that they get to that level of detail though when you're in the vegan world. So
cauliflower or something like that.
Cauliflower, good. Yeah.
Perfect.
Jackfruit,
It still sounds good to me.
Yeah. It's the sauce that really does it. So
yeah, exactly.
The buffalo sauce.
Well, thank you again so much. We really appreciate your time.
Thank you very much.
Bellringer is a podcast by Invest Buffalo Niagara, the region's privately funded, nonprofit marketing and economic development organization. Please rate this podcast, follow our social media channels, and read our blog at buffaloniagara.org for the best of Buffalo Niagara. Come grow your business with us.